This is the lecture on the evolution of diversity. So when we think about the diversity of living organisms on the planet and the history of the planet and how they got to be here, there are several major transitions that need explanations. The first is the origin of life. So abiogenesis. How do you get a living organism when there were no living organisms before? Now, I want to indicate to you first that the Earth is very, very old. So not just geologists, but from a wide variety of natural sciences, research has come together to estimate the age of the Earth at about 4.5 billion years. So these are geologists, they're astrophysicists, etc. that come up with this value. And if we track the early history of fossils, the earliest evidence is about 3.5 billion years ago. So a big question, how did the first living organisms arise? Well, what we do know is that the early atmosphere had no oxygen. It had a lot of hydrogen gas, ammonia, methane gas, and water vapor. And in the 1950s, Miller and Urey set up some experiments to test what was called the primordial soup hypothesis. And the hypothesis was this, that with a few of these simple ingredients that were known to be on the Earth's atmosphere at that point in time, could you build chemical structures that would lead to living organisms? So they had a really neat little experimental setup where what they basically did was they had all of the gases in a chamber that had electrical sparks. They then ran that through a closed circuit that had some water that was heated. So you've got heat, water, and electricity. They let this system run for a couple of days and found after a few days that in the water were amino acids, purines, and pyrimidines. Purines and pyrimidines are your nucleic acid bases. So we were talking about things like adenine and thymine. So we've got some biological type molecules that were in this water. Now remember, in science you always have to update and rethink your ideas when you get new information, and new information that would change how we would perform Miller and Urey's experiment today is that the early atmosphere also had carbon dioxide, hydrogen sulfide, and sulfur dioxide. So if you rerun those experiments, what do you find? Well, you get even more diverse chemical products possible. So this is not necessarily proving that you can have life formed from no life, but it does set up the circumstances where you can get biological type molecules arising without any living forms. There is a huge amount of research being currently done by groups like Craig Ventnor, where they are researching the construction of synthetic or artificial life, and that helps us to understand how early life would have formed. So for example, we know that it's likely that RNA was the first genetic code, and from that DNA evolved. We also know that it's fairly easy to create cell membranes. Many of you young ladies actually may use a product called micellar water to wash your faces with. And what they basically do in that is they take some phospholipids that are shaken with water, and it creates little micelles, which are basically bubbles with oil in them. And a micelle structure is similar to what you would need in order to create a very simple cell membrane. Once you've got a cell membrane, you've got inside and outside. We've also got your amino acids, and you've got some purines and pyrimidines. So you can see how we're starting to acquire all of the basic ingredients that would be necessary to create a living cell. The next big transition in the history of life is the evolution of the eukaryotic organisms. These are organisms that have membrane-bound organelles. They've got a nucleus, chloroplast, and mitochondria. Now, there are three main domains of living organisms. There are the bacteria, the archaea, and the eukaryota. So bacteria, you've experienced earlier in the semester. These are single-celled prokaryotic organisms. You looked at the cell structure of one of them. Archaeans are very similar. They're single-celled, they're prokaryotic, but these are the extreme single-celled prokaryotes. They're the halophytes. They're things that live in really, really salty conditions, really hot conditions, really acidic conditions. They're the methanogens that make methane gas. So they're quite odd. Eukaryotes, on the other hand, the domain eukaryota has got animals, fungi, plants, and then a variety of other lineages that we call the protists. Now, what do they all have in common? They all have a mitochondria, which is an organelle that's used for energy production. And plants have got an additional organelle called a chloroplast that helps with photosynthesis. So how did eukaryotes arrive? The theory of endosymbiosis is the explanation for how eukaryotic organisms arose. So you could think of it this way. If a prokaryote engulfs another prokaryote but does not actually digest and eat it, you can set up the situation where now you've got a eukaryote, because maybe prokaryote 1 ate prokaryote 2 because prokaryote 2 could perform a certain function like photosynthesis. And if you don't eat prokaryote 2, then you're carrying within your own body the ability to photosynthesize. And it's now understood that most of the organelles in eukaryotes have derived from prokaryotes, such as mitochondria and chloroplasts. And there are many lines of evidence for endosymbiosis. One of them is that mitochondria have their own membranes, just like a bacterium. They also have their own DNA that's in a simple circular genome, so you can actually trace your maternal lineage through mitochondrial DNA, because it turns out that humans get their mitochondria from mum's egg. Dad's sperm is too small and does not carry any mitochondria in it, so it doesn't provide any mitochondria to the zygote. And your mitochondria reproduce by binary fission, which is the same method that bacteria reproduce. So here's a nice living example, a clue as to why endosymbiosis occurred. There is a eukaryotic protist called Paramecium bursaria, and it eats blue-green algae. But instead of digesting and eating the algae, it holds the algae in its cell and it moves them around in patches of sunlight. So the Paramecium bursaria provides the algae with a nice place to grow, and it provides them with transportation to sunlight, and the algae in return produce food for Paramecium. So it's a mutually beneficial interaction. So now let's talk about the origin of species. Species means kind or appearance, and speciation is the formation of new species and is one of the primary points of focus for macroevolution. So there's sort of two branching patterns, so contrasting patterns. The one is a branching pattern and the other one is non-branching. So if you imagine back to population genetics and you think of your simple population, and over time you can accumulate mutations and undergo genetic drift, and you can undergo gene flow, and over time your population can morph from one form to the other. This is called non-branching evolution. And that really, you can very easily describe non-branching evolution with all of your microevolutionary forces. But in order to get the diversity of organisms on the planet, you really need to ask the question of how do you get new species branching off from a parent species, so branching evolution. Now, the long straight trajectories of once the populations have been divided into two and then accumulating different mutations and forces of selection, etc., you can imagine those through microevolutionary forces. But precisely what is happening at the branch point, what divides a population into two that precipitates macroevolution, and that's going to be the focus of this lecture. So let's first talk about what a species is. We use the biological species concept, and that defines a species as a group of interbreeding natural populations that are reproductively isolated from other groups. So what that means is that there's something that's preventing you from reproducing with other individuals that it's found in nature. So, for example, you can have a lot of similarity between species. So here's the eastern meadowlark and the western meadowlark. They actually don't mate with each other. They have different courtship signals, different songs, and they are reproductively isolated from each other. On the other hand, you can have a huge amount of diversity within a species, like in the human species Homo sapiens. Individuals can vary dramatically in their appearance. However, we are not reproductively isolated from one another because you can meet and marry and reproduce with somebody of any phenotype. So, macroevolution, the evolution of new species. The biological species concept stresses reproductive isolation, so we need to talk a little bit about what that means. There are two main categories for reproductive isolation. There are barriers that prevent mating between species or prevent fertilization if mating is attempted. So it's before the formation of a zygote. The second category is post-zygotic barriers, and these are barriers to reproduction after fertilization has occurred. So I'm going to give you a few examples of each, starting with pre-zygotic barriers. So one of them is habitat isolation. If you live in different habitats and rarely meet, you're not going to be mating. So garter snakes, there's an aquatic form and a terrestrial form. They both live in the same area. They, in nature, don't mate and meet, therefore they're different species. Behavioral isolation. So those meadowlark birds have a form of behavioral isolation as well. It's when males and females don't recognize each other as potential mates. So in lacewings, there are many species of them. They all look very similar, but they produce and exchange courtship songs with each other, and those courtship songs indicate the correct species. I'm going to play a couple of them for you. You can also click on these and listen to them. So that's Carnea. Pallida sounds a little different. This is Agilis. And then there's Leucocina. And Mediterranea. If it's not very loud, you can step through the PowerPoint yourself and just click on the bells. Now, in each of these species, the male will perform the song, the female has to hear the correct song, then she has to perform her song back, and if they have the correct signal between them, they will then mate. Temporal isolation is when you breed at different times of the day or seasons or years. This is found in plants, this is found in animals. Many plants only flower or blossom at certain times of the year. Just think about cherry blossoms in Washington, D.C., or Palo Verde blossoms in Arizona. The skunks, there's a western one and an eastern one. Their ranges overlap, but they mate at different times of year. The timing of when they mate has got everything to do with food availability for their offspring once they give birth. So there's some natural selection for when you're going to be breeding. Mechanical isolation, this one gives a lot of people some giggles. This is when species are anatomically incompatible. So to put it mildly, tab A does not fit into slot B. For many organisms where there's direct deposition of sperm, you need the male and female genitalia to coordinate appropriately so that you can deposit sperm. Gametic isolation is when the gametes are incompatible. Contrary to what you think, the sperm doesn't just go ahead and burrow into the egg. It's actually an interplay between the egg and the sperm, and the egg will only adhere to sperm that has the appropriate proteins that are being expressed on the surface of the sperm's membranes. And as you know from the central dogma, if you have a single mutation in your DNA sequence, you may end up with a single mutation in your amino acid sequence, and that could result in a protein with a different three-dimensional structure, and that might actually result in gametic isolation or incompatibility. So post-zygotic barriers are what would happen if the hybrid between two species fails to develop into a viable fertile adult. And there's a variety of different mechanisms. There's reduced viability, so the zygotes don't develop properly or they don't reach maturity. There's reduced fertility, where the hybrids grow to adulthood but they cannot produce sperm and eggs themselves. And then there's hybrid breakdown, which is when the offspring have reduced viability or fertility. Some examples of post-zygotic incompatibility include differences in chromosome numbers between species. You probably know about the example of a horse and a donkey breed forming a mule. The mule will likely be sterile. But the same thing can happen with sheep and goats. Sheep have 54 chromosomes, goats have 60 chromosomes. And very rarely are they ever successful in producing an offspring. So to understand speciation, you have to ask the question, this key event, what happened that caused your single population to become or to start becoming two separate populations? What are the mechanisms that make one species become two? And there's two modes of speciation. The first is called allopatric speciation. The second is called sympatric speciation. In allopatric speciation, there's geographic separation. A mountain forms, a river forms, a canyon forms, continental plates drift apart. So you're separating your population into two. And when your population has been separated into two, each one of those subpopulations then undergo different evolutionary trajectories in that they're accumulating different mutations, different genetic drift, different gene flow, different natural selection, and over time they can go and differentiate into two species. The second mode is called sympatric speciation, and is a type of isolation that can occur without geographic isolation, and so there are some other modes for how that can happen, and I'll get into that in a second. But in allopatric speciation, you fragment your population into two or more isolated populations. And the fossil record is really helpful. It's an archive for us of macroevolution, and we can actually time the evolution of new groups to when continental plates separated and moved apart. The geologic record is extraordinarily helpful for helping to reconstruct the history of the diversity of life forms. So, for example, about 250 million years ago, we had a supercontinent called Pangaea, and there were many extinctions that occurred, and these mass extinctions resulted in a reduction in life forms, and then nature abhors a vacuum, and so what happens is you end up with the surviving life forms end up diversifying, and you get multiple instances of adaptation. Now, speciation only occurs with the evolution of reproductive barriers, so if your isolated population comes back into sympatry with your main population, and they can still breed, we would say that speciation hasn't occurred. However, if populations have been separated for long enough that they don't interbreed, then we would say that speciation has occurred. Now, sympatric speciation is a little different. It doesn't require geographic separation. And there are three common modes. One is called autopolyploidy, the other one's called allopolyploidy, and the last is sexual selection. In autopolyploidy, it's a bit of a mouthful, you have errors that happen during meiosis. So this is non-disjunction. And that can result in offspring that have extra chromosomes. So imagine that when you were going to try to make a sperm or an egg cell, instead of the chromosomes dividing from 4n to 2n and then from 2n to 1n to make a haploid gamete, you end up with a diploid gamete. You may then have fertilization of a diploid gamete, and that would create a polyploid offspring, so an offspring that's got many copies of the genome. And in some cases, those offspring are viable and can survive, but they cannot breed with individuals that have the same ploidy as their parents, so they would be considered a new species. This happens in nature, but it actually also happens in agriculture. So we deliberately treat some crop plants with a chemical called colchicine. You may know it as a treatment for gout in humans or high uric acid in humans. But if you treat a plant with it, it creates errors in meiosis when the plant is making pollen. And so you can create triploid crops, 3n, tetraploid, 4n, hexaploid, 6n, octoploid, 8n, etc. And many of your common fruits and vegetables, it's likely you've had an apple or a banana recently, maybe you're wearing cotton pants. These are all created by autopolyploidy. So in this case of autopolyploidy, you have extra copies of the genome, but all of the copies of the genome are coming from a single original species. That's auto. So it's to yourself you make polyploidy. That's what autopolyploidy means. Now the second form is allopolyploidy, and that's when you have genomes from different species fusing to create a new species. And one of the most famous examples of this is called the triangle of U. And in this you have three ancestral species of brassicas. These are wild mustards, and when they crossed with each other, they formed three new species of hybrid mustards. Some of them include canola oil, etc. So if you want to read more about it, definitely look up the triangle of U. Now, sexual selection is very important for sympatric speciation. And I want you to think about this from a theoretical perspective. Imagine you've got a population. Males have many different colors. Females show no color preference for mates. They'll breed at random with each other. So what that means is that a female's not choosing which male she wants to mate with, so her offspring could be any color. She has no preferences. Now, imagine a mutation arises in females where females with the mutation really like blue males. What this means is that females who really like blue males, their sons are more likely to also be blue, and their daughters will likely carry that same mutation, preferring blue males. So within one step, you have a situation where females who like blue males will have blue sons and daughters who like blue males, and the rest of the population will continue in the same way it ever has. So you've basically created two subpopulations within the main population. Now, over time, you would have to have the accumulation of other mutations and other barriers to reproductive ability, so something like temporal isolation or behavioral isolation, to really cement that speciation process. Do we have real examples? Of course. So there are these little tiny fish that are found in Africa. They're called Pandamelia. There's two species. There's Pandamelia pandamelia, which is the one on the top, and Pandamelia nererii on the bottom. Now the males are very beautiful. The Pandamelia pandamelia males are blue. The nererii ones are rainbow colored. But if you put them in monochromatic light and you let females choose which one they'd like to mate with, the females can't tell the difference between the two species and will mate indiscriminately in monochromatic light. Whereas with normal full spectrum light, they'll only mate with the appropriate male. What this tells us is that this speciation process is very young, and it's not yet solidified where there are strong barriers to reproduction. So what about the speed of speciation? Well, traditionally it was viewed as this gradual divergence, and you'd have intermediate fossils representing each little step from one to the other. And while this can be found in some groups of organisms, in other groups of organisms we couldn't find intermediate fossils, and that was a point of contention because you would expect to find them. We have another model for macroevolution, and that's called punctuated equilibrium. And this is where you go for eons with very little change, and then you'll have a massive amount of change happening in a small amount of time due to some very strong mutation. We find this in other groups of organisms. We have very strong evidence this can happen. So, for example, if you end up with some changes to your Hox genes that change the shape of your limbs, that can happen with a very small number of regulatory changes to change from a long arm to a short arm. So neither gradualism nor punctuated equilibrium is correct, or the only tempo for macroevolution. There are examples of each tempo in nature. Now, when we have this huge number of organisms, we need to be able to classify them or organize them. And the form of organization that we use is called the binomial nomenclature. It was originally proposed by Carolinus Linnaeus. He was a Swedish botanist. And each species is given a two-part name. So this two-part name has got the genus name and the species name. So in this example, Pardus is the species name, and Panthera is the genus name. And we always italicize the scientific name to indicate that it is a scientific name. So every organism that has been described so far has been given its proper binomial name. And the goal of classification is to reflect both the phylogeny, the evolutionary history of the species, and to provide it with something that we can use to identify it again. So, for example, in the order Carnivora, so these are mammals that eat meat. There are multiple different families. There's the feline family, so these are cats. Mustelids, these are musty-smelling ones. And canids, these are the dogs. So within the felids, you've got Panthera, so that's a genus that's got panthers in it, such as the leopard. You also have your domestic cat as well. In the mustelids, there's two different genera. Mephitis and Lutra are some examples of them. You've got your skunk and your otter. So skunks and otters are more closely related to each other than they are to leopards. And then in the canid group, you've got the genus Canis. That's got Canis lupus, which is the wolf, and Familiaris, which is the domestic dog. So obviously in this representation, the domestic dog and the wolf are more closely related to each other than they are to otters or to the leopard.